This is part 2 of the recorded lectures for cellular structure. Let me get this slideshow going. So we were looking at organelles within the eukaryotic cell and the Golgi apparatus, or the Golgi complex, is an organelle that's its function is receiving lipids and proteins from the endoplasmic reticulum, and then taking those products from the endoplasmic reticulum, sorting them, tagging them, maybe tagging them with extra chemical groups or extra chemicals, then packaging them into a vesicle, and then distributing them either within the cell or outside of the cell by exocytosis. So the Golgi apparatus is a series of flattened membranes, and in plant cells, it also makes polysaccharides, like converts glucose to starch, for example. All right, vesicles are membrane-bound sacs that function in storage and transport. Their membrane is a phospholipid bilayer like the plasma membrane. So a vesicle can fuse with the plasma membrane and then release its contents outside the cell. Vesicles can also fuse with other organelles. For example, if you have a vesicle that's storing food particles, it can fuse with a lysosome containing digestive enzymes. And when they fuse, those digestive enzymes will break down the food particles. Animal cells have a centrosome, which plant cells do not have, and the centrosome consists of 2 centrioles. And centrioles have 9 groups of 3 microtubules each. So 1, 2, 3, 4, 5, 6, 7, 8, 9. And then it's a 9 plus 2 arrangement. So I don't remember what the plus 2 is, but anyway, there's 9 sets of groups of 3 or triplets of microtubules. So centrioles are made of microtubules, and their role is in cell division. Lysosomes are organelles that contain digestive enzymes. They're only found in animal cells, and they break down solids, food particles. They break down worn out cells that need to be recycled. Their function is breaking down chemicals. All right, chloroplasts are only found in plants, and they carry out photosynthesis. Like the mitochondria, they have their own DNA and ribosomes. And they contain the pigment chlorophyll as well as other pigments. Vacuoles are larger than vesicles. They function in storage and transport. But the difference in a vacuole and a vesicle is that the vacuole cannot fuse with any other membranes. And the central vacuole in a plant is very large, and it stores water, and which is a difference between plant cells and animal cells. The cytoskeleton in the cell is the network of protein fibers that help maintain the cell's shape, hold the organelles in their places, and allow the cytoplasm and vesicles to move within the cell. And it enables cells within multicellular organisms to move. So the types of fibers are, the thinnest fibers are the microfilaments, and you find them on the outside part of the, right up close to the plasma membrane. So if these are your microfilaments, the plasma membrane would be here. I'm drawing it very badly, but That would be your plasma membrane. The microfilaments are in the cortex, which is the outside of the cell, the closest part to the plasma membrane. The microtubules are the largest protein fibers. They are found in the interior. They help maintain the cell shape, and they also are used to form spindle fibers when the cell is dividing. They are the thickest of the protein fibers. And then intermediate filaments are found throughout the cell, and they hold the organelles in place. A little bit more about the microfilaments. A microfilament called actin is found in muscle cells, and it helps muscles to contract. And there's a little bit more information on intermediate filaments, and then microtubules are the thickest, and they are called spindle fibers when they function in cell division. They actually function in pulling replicated chromosomes to opposite ends of a dividing cell. So they attach to the chromosomes and pull them apart so that each new cell gets a copy. Flagella and cilia are structures that permit movement. They're located on the outside of certain cells. All cells do not have flagella and cilia, but flagella is a long whip-like tail that you find on a sperm, for example. And cilia are shorter little hairs, and they, in some single-celled microorganisms, they help the organism to move, but in humans, cilia is used to move some kind of substance or particles across the surface of the cell. So for example, there's cilia that lines the respiratory tract that beats all in one direction to help move the mucus and trapped pathogens and possibly harmful substances up and out of your lungs, so they don't go deep in your lungs and cause an infection. So flagella is a long tail, and cilia are short hairs. All right, here's the plasma membrane, which the next chapter will go into more detail with it. But both types of cells, prokaryotic and eukaryotic, have a plasma membrane. Its primary structure is that it's a double layer of phospholipids, and the large blue molecules are proteins. You can also see this is cholesterol can be found within the lipid bilayer, and carbohydrate chains can be attached to the phospholipids, and we call them glycolipids. And carbohydrate chains can be attached to the proteins, and in that case we call them glycoproteins. Microvilli are folds in the surface of a cell. So microvilli help to increase surface area. If you have, for example, if you do a cross section through the intestine, if this is the lining of the intestine compared to this one, that the inside is folded, which one has the greatest surface area for absorbing nutrients into the walls of the intestine and then into the blood? And that's going to be the one with the folds. So in the intestines, they have folds called villi, and then those villi have additional folds called microvilli. So their purpose is increasing surface area for the absorption of nutrients or for other chemical processes. Plant cells are connected with each other by plasmodesmata, which are channels that pass between cell walls of adjacent plant cells. They're different from the gap junctions in cells because gap junctions are similar, but they connect plasma membranes instead of cell walls. And we're going to talk about gap junctions now. Well, it just has a little bit about them. So Animal cells are connected to each other by tight junctions, desmosomes, and gap junctions. Gap junctions have a gap or a channel between the two cells, allowing materials to flow from one cell to the other. Tight junctions form a very tight seal, like in the urinary bladder. The tight junctions prevent leakage of urine into the rest of your body. And desmosomes are very strong attachments at one point. They're almost like a spot weld. They're very, very strong at one spot. The extracellular matrix holds cells together. So the extracellular matrix tends to consist of protein fibers like collagen, and then some type of a fluid or solid matrix. Gap junctions, we just talked about this, but you can see them right here. You can see these are the gap junctions, and then you can see it, how the gap junctions connect two plasma membranes to each other. And then there's a gap that goes all the way through each cell. So ions can be transported, nutrients and other substances can be transported through these gap junctions. The cilia that all move in the same direction, they are coordinated to where they're all going to move in the very same direction at the very same time. So like if this was one of our cells and it had cilia on it, they're all going to be moving this way together at the very same time. And that coordinated movement is possible because they're connected by gap junctions. A tight junction is a watertight seal between two cells, and it prevents leaking. I gave you the example of the urinary bladder. And a desmosome is a very strong connection that is especially found in tissues that stretch, like the skin, the heart, and muscles. And that is the reason why if you've ever had a sunburn, your skin gets shed and you can pull off sheets of skin because the skin cells are actually connected by desmosomes. And another example is like dandruff. So if you have flaky or ashy skin, those flakes of skin are not one skin cell. We can see the flakes of skin, so they're composed of many skin cells. You know, so your skin does not shed in individual cells. It sheds in sheets. Okay. Cell walls are found not in animal cells, but they're found in every other type of living thing. They're found in the bacteria, prokaryotes, and the chief component in the cell walls of prokaryotes is peptidoglycan. In plants, the cell wall component is cellulose, and in fungi, it is chitin. The cell walls are composed of chitin, which is CHITIN. This is a table listing the component of the cell and whether it's present in prokaryotes, animal cells, or plant cells. It's really important to understand that the structures that are present in all cells are when you have yes on all three columns. So cytoplasm, ribosomes, and that's it on this table. Okay, if you keep going, let's see here, cytoskeleton. Sometimes flagella, sometimes cilia, because different ones have, these structures to help them move. And the plasma membrane is found in all. So everything I've circled is found in all types of cells, whether they're prokaryotic or eukaryotic. And then you can go through and look and make sure that you know which structures are found only in animal cells, not plant cells. And that's going to be your lysosomes, trying to make a check in the box. Okay,